no comprehensive evaluation of this statistical method to detect the changes in M6A. So there are many functions to call the peak. People like MAG or this exon peak or other peak calling functions, but they are all using the statistical method, but it's a little bit difficult to evaluate different functions to say that which one is better or which one is not. So those are the most widely used technology, and to overcome this resolution issue, single base M6A sequencing technology has been developed so far. And there are many different type of technologies based on their key method, and they have slight issues on their own. So mainly it can be the low coverage issues, and also it can be a high false positive issues depending on the type of method. So we want to determine that actual intrinsic M6A site. So we first tried to collect the data first, and then we decided to use MARISIC data. And there are so many variable M6A data existing in public. So we have to decide what MARISIC data we have to use in order to find the intrinsic M6A site. So we first examine the public data. So the majority of data in the GEO are the cell line data, and the primary cell line data is very limited among these deposited cell line data. But what we want to identify is intrinsic M6A site that are methylated in normal physiological conditions, so that the perturbed cell line cannot be used to explore the intrinsic M6A sites in our case. So we need the normal human tissue data to investigate the intrinsic M6A site. So to meet our hypothesis, we actually collected the human tissue MARISIC data used in two different studies, which is we just call them one as NGDC data set, another one we call them CBBC data set, just based on the data source. So we eventually collected 69 human tissue MARISIC data from 24 different tissue types. So NGDC data consists of 7 donors and 23 tissue types, and CBBC data consists of 7 tissue samples from 5 donors. And these are the overview of the sample, that each tissue conditions, mostly NGDC samples are there except for the CBBC data, because CBBC has only 7 tissue types. But we integrated both of them, and then we we aligned the sample to the human genome, and then predicted the peak region using the exon peak function. And I think it's not. I think it's not important, so let's just skip it. But I just processed all the sample in the same pipeline I developed, and that's all. And then we processed the sample in the same pipeline, and then predicted the peak using the exon peak 2, and then after the peak calling, we filter out the peak to find the more high confidence M6A site, high confidence M6A peak. So these are the final peak calling result. And as you can see here, 2 data set has quite different number of predicted peaks across the samples. And then all the tissues are also have a high variability across the tissue types. So after we predicted the peaks across these tissue samples, we actually observed the predicted peak region across the samples, and the top 7 is the CBBC sample, and the bottom samples are the tissue sample from the NGDC data. And then we can see that you can see that even though this high variability between the data set and then high variability across the tissue types, we were able to see that exactly the same region on the gene was have these same predicted M6A peak across different tissues or different sample types. So we were able to observe these extensively shared M6A peaks across data set. And like I briefly mentioned, there are intrasample and intersample variability in our data set. And in addition to that, the most important limitation is these are meristic data. So this is not a single base data, so that we cannot pinpoint the exact M6A size across this predicted peak region. So let's say this region is predicted as peak, and this region could be very small, or could be 100 base pair, or sometimes 1,000 base pair alone. And across them, it's a little bit difficult to exactly pinpointing the single M6A size. So the next question is, now we collected all the normal human tissue meristic data, and we predicted the M6A peak, and then we successfully see that those share the context-independent M6A peaks, but still we don't know where exactly the M6A site. So we try to utilize the single base M6A detection data. But issue with those single base data is still the technologies has high false positive issues with the single resolution data. For example, 39% of reported M6A size are detected by MyClip or MyClip2, which are known to have high false positivity, and some high precision or high accuracy sequencing data has still have a very low coverage issues. So if we examine these samples, so these are the cell lines and the different single base technology, and then these are the detected M6A genes by this different single base technology. And if we examine the jointly reported genes by different method, we can see that if we just increase one more sequencing technology to detect the same gene in the same cell line, the number is significantly dropped, and then the number is somewhat similar up to 4 or 5 number of methods to detect the same number of genes. And over this number, the jointly reported genes is drastically dropped. So what we can see here is, if we rely on just one single base technology, we may ended up with very high false positivity that cannot be detected any other different technologies at all. So what we did is we first collect the entire reported single base M6A size in public, and then we require a site to be detected by at least 2 different sequencing technology, except MyClip or MyClip2 combination, because these 2 technologies are known to have high false positivity. So if a site is only reported by MyClip or MyClip2, then that site is not the high confidence site. So these are the total number of collection we retrieved from M6A Atlas 2 sites, which comprise 13 different single base technology across 24 cell lines, across 28 experimental conditions, and across these over 3,000 different genes. So as you can see, the number is really high. So approximately 480,000 single sites are reported as M6A sites in different conditions or cell lines. And if we applied our criteria, then we only have this number. Only 26% of total collection are past this criteria, and then we decided to use these high confidence sites only to map these sites to the M6A peak region. So if we briefly overview the selected sites, then the overall regional proportion across gene region is pretty much similar. What we know is more enriched in the 3' UTR region. And their regional database are pretty much similar. And if we actually validate, after we select those high confidence sites, we validated these sites, whether they are biased to some specific region or they are actually more robust than those excluded sites. And if we compare these validation results, we were able to see that the selected sites versus excluded sites, we were able to see that their regional proportion were pretty much similar between 3' UTR exon and 5' UTR region. So there was no regional bias between the selected versus excluded sites. And also, we are able to see that the majority of the selected sites were reported more than 2 studies or more than 3-4 studies versus those excluded sites. Only 96% of those excluded only reported in 1 study. That means it hasn't been reported at all. Even though they use the same sequencing technology. So the reproducibility of those excluded sites were extremely low in general, and the reproducibility across cell lines or conditions were also extremely low in those excluded sites versus the selected sites. So the conclusion is, even though we applied the threshold ourselves, but still they have no regional bias and they are more robust than the excluded sites in general. And in addition to those overall examination, we also actually statistically overlap those sites, that the excluded sites and selected sites, that whether they are having more M6A peaks versus more without the M6A peaks. So the top panel is showing the number of M6A peaks that overlapped with these excluded sites versus selected sites in each data set respectively. And we can see that the M6A peaks M6A peaks is significantly have more selected sites than the excluded sites. And reversely, if we compare the M6A peaks without any known single M6A sites, then we can see that the excluded sites versus selected sites, then we can see that the M6A peaks without an M6A site is significantly higher when peaks or compared with the excluded sites together. So overall, we are able to see that our selected site has higher confidence than those excluded sites. So after with this validation, now we have those extensively shared M6A peaks and those high confidence M6A sites. So with these two, we map those high confidence sites and the shared M6A peaks. So we overlap those predicted peaks across tissue data and the selected high confidence sites together, and then we compare the peak locus overlapping result. So some sites has no overlapped peaks versus some site has overlapped peaks there. So by overlaying with the M6A peak from normal tissue conditions and selected high confidence sites across 28 different experimental conditions or 24 different cell lines, we were able to refine ourselves that the high confidence M6A sites that existed in the normal tissue conditions together. So as our definition, intrinsic sites are the sites that context independently methylated in normal physical conditions, and to meet those definitions, we overlap this peak from tissue data and this single size from the high confidence data. So and after this overlapping, we were able to find the size that overlapped with the M6A peak in each different data set. And then still the NGDC size or have higher average number of a peak than the CBBC size. So as the overlapped M6A size with peaks also higher in NGDC data. And as total, we were able to identify these number of total unique M6A sites in each different data set. And these are the predicted, these are the overlapped size across the different tissue samples. Note that these tissue sample with this star mark is the single sample tissue. That means there is only one tissue sample in NGDC data only, and without any other sample in the same tissue type. So if after we overlap, we were able to pinpoint these specific sites. So the thin bar on the top is the predicted M6A sites, and then if that are overlapped with M6A peaks, so we can see that there are predicted peaks here, and then if these are a little bit difficult to see because of the scale, but if we zoom into this specific region, we can see that there are enriched peak region there, and then there is a single M6A size over there. So we were successfully identify context-independent M6A size that shared across 24 tissue types. So after we complete this overlap, we want to identify the real intrinsic size. So if we examine the overlap distribution across the size of M6A size across samples, we were able to see some kind of saturation if we increase the number of samples, and we see some very extensive overlap if we increase the more samples in NGDC data. That means that there are a specific portion of M6A size that are shared across almost the entire samples. And this distribution is not random. So if we permit the overlaps and then plot the distribution, then distribution is completely different than what we exactly observe versus just the background distribution. So what we see here, that this high number of samples that are overlapped across the entire samples in each data set are very unique distribution. So we compare these two distribution from the data set and the background distribution. We classify the size that pass the threshold FTR 0.01 as intrinsic versus the overlap less than 15% out of the entire sample as the infrequent size, and then just in between them we just name them other sites. So in NGDC and CBBC dataset, 16.3% and 22.9% of sites were identified as intrinsic sites in two data sets respectively. And if we examine this intrinsic size, that how many tissues they are overlapped each other, then in the CBBC dataset, 95% of intrinsic sites were existed in all 7 tissue types in CBBC, and only 5% were overlapped in 6 tissue types. And I believe this 5% is mainly due to the sample quality of a couple of samples that has a little bit have low coverages and etc. And in NGDC dataset, we were also able to see that the 63% of these intrinsic sites, or let's say significantly shared M6A sites, FZR less than 0.01, is overlapped across entire 23 different tissue types, and the minimum was 18 different types. And as I briefly mentioned, there are 7 tissue types that has only one samples. So if a peak was not able to predict it in that those single tissue samples, then due to some noise or antibody issues or whatever, some biological variations or technological variations, we just lost those sites. And if we account those limitations, still these numbers are really high. That majority of the intrinsic sites we identify are in general shared most of the tissues we have in our data set. So from those significantly shared M6A sites, we further refine ourselves to overlap those significantly shared sites from CBBC and NGDC, so as we were identify these 5,945 M6A sites that exist in both data sets. That means these sites were existed in 7 tissue types in CBBC and also the 23 different tissue types in NGDC at the same time, even though these two data sets has higher intersample and intrasample variability between the data set. And these sites are technically shared in 1,384 genes across 24 different tissues. So yeah, it's a little bit confusing, but 6 tissues are in CBBC are also in NGDC, but the kidney tissue are only in the CBBC dataset. So if we overlay these two datasets together, that we have a total of 24 tissue types across 2 datasets. So finally, the intrinsic M6A sites we identified that is first identified the significantly shared size and then overlay the 2 datasets together, so as we finally have this number of high confidence intrinsic M6A sites deposited in 24 tissue sites, 24 different tissues across out of 69 tissue samples. So after we identified the intrinsic M6A sites, we try to figure out the distinctive feature of these identified sites compared to their counterpart, which is the infrequent site. So what we are comparing is intrinsic size that is context independently shared M6A sites versus infrequent sites that are very sparsely shared. So some of them are tissue specific and some are shared in only a couple of tissues only. So if we compare the sites with the infrequent and background together, we were able to see that intrinsic sites are significantly enriched at the 3' UTR region. So the enrichment around 3' UTR region of M6A is one of the known feature. So in majority, in general, M6A sites are known to enrich around the stop codon region. However, the enrichment of intrinsic sites is much more significant compared to both infrequent and background. Very interestingly, intrinsic sites also tend to have higher methylation levels than the background or the infrequent sites, and also tend to have a little bit more conserved than the others based on the fast sequence scores of each sites across intrinsic versus infrequent or the background. And what we see here, in general, infrequent and background are pretty much similar, whatever the methylation level or their method, like a genomic distribution or the methylation. But the infrequent sites are different than those two. And another interesting feature is intrinsic sites are more clustered than each other than other type of sites. So if we compare these figure, if we compare with the infrequent and background, what we see in general is the number of sites per genes are it's kind of much higher in the background genes versus slightly higher for the intrinsic than the infrequent. So if we see that the number of sites per gene, that means the average number of M6A sites per gene. And if we compare the average adjacent M6A sites within this specific window size, what we see here is whatever the window size, intrinsic size tend to have more adjacent intrinsic size than the other type of infrequent or background M6A sites. And the previously published data which is Glory, one of the high confidence single-base technology, also reported some specific type of M6A site that are known to cluster each other, and those number of clustered sites are also more enriched among these intrinsic sites than the infrequent or background sites. So altogether, in general, intrinsic sites are more clustered than the other type of M6A sites. And this is another feature that intrinsic sites have a tendency to reside at the shorter and also tends to be the last exon of the gene, which potentially prevents the M6A inhibition by EJC. So this concept is a little bit new, but the overall concept is if the exon length is longer, then there is higher probability that is bound by exon junction complex, which prevents the M6A methylation around the sites. But what we see here is intrinsic sites are resided at the relatively shorter exon than the other infrequent or background sites. And those exons where intrinsic sites is relatively less exon than the other infrequent or background sites exons. That means their location is relatively in the last exon, and also those last exons are relatively shorter than the other, so that the bound by EJC is prevented due to the exon length, so that the M6A methylation inhibition by EJC is prevented, so the M6A can be methylated or less suppressed by EJC than the other infrequent or background sites. I believe, so this is the last feature, that is a sequence level feature comparing the infrequent sites and intrinsic sites. So this is analyzed by Tinga, which is he fine-tuned his M6A BERT model to classify the sequence from upstream and downstream 150 base pair from the intrinsic size and infrequent size, and then this classifier classify the sequence, whether it is intrinsic sequence versus infrequent sequence. And what we see here is, in general, they were able to classify the intrinsic M6A sequence between the infrequent sites with these performance. Also, the intrinsic site sequence was able to be classified compared to the background sequence. So what we can infer from this data is intrinsic sites have a distinctive neighboring sequence than the other infrequent or background sequences. So so far we identified the intrinsic size and then analyzed the feature of the intrinsic size compared to their counterparts. And the question is, okay, we identified and then they have distinctive feature than the infrequent size. Then how they can be methylated context independently? That's the next question. So how are these intrinsic sites are deposited, which is we want to investigate the upstream regulation of intrinsic M6A sites. So these are the methylation heat map that we can see that the columns are the different tissues and the rows are the intrinsic M6A sites. And you can see that it's pretty much consistent methylation, what we can see here. So this is also very interesting feature. So they are not only existed across all the tissue types, but also their methylation level are also very consistent across tissue types. But the range of the methylation level across the M6 intrinsic sites are very different in a level of log 2 from 0 to 15. So the scale is quite different. So I analyzed the cosine similarity of the methylation level across the tissue types between intrinsic versus the permuted methylation level. And what we can see here is the intrinsic sites are significantly higher cosine similarity of methylation level across tissues. And also the standard deviation of methylation across tissues of intrinsic size compared to the background, we can see that it has a significantly lower standard deviation of methylation across tissue type. So very interestingly, intrinsic sites are methylated across 24 tissue types, and also their methylation level is significantly consistent across tissue types. So however, their methylation range across the sites are very different. So what we initially speculated is maybe there are different sequence motif that methylated these different sites. So, for example, higher methylation level site has DRSH motif, but the lower methylation cluster maybe have different motif, so that the main methyltransferase or main writer proteins are bound to higher methylation level cluster versus low methylation cluster may be methylated by some other different type of writer, which is not known as the major writers. However, what we observed is it doesn't matter. All the intrinsic sites has the same DRSH motif. So we can see that 92% of intrinsic site have exact DRSH motif. And we examined the infrequent and background sites also. And even the infrequent or background site has the same exact DRSH motif in 96% and 95% respectively. So if we analyze the statistical motif search, then intrinsic and infrequent and background has exactly the same ERACW motif, which are slightly different than the known DRSH motif, and they are all significantly enriched across all type of the M6A sites. That means the main writing mechanism or main methylation mechanism across intrinsic, infrequent, and background is similar. There is no difference. So the main methyltransferase, so methyl-3 and methyl-14 complex will be the main upstream mechanism. So the next question is, okay, the main methyltransferase will methylate these sites, but why? why only the intrinsic sites are methylated across all tissues, and why some has higher methylation versus some has low methylation, even though the same methyl 3 and methyl 14 complex will methylate these sites. So we expanded our analyze to the neighboring hood, or so-called the cofactor, that is known to help methyltransferase complex to methylate or demethylate those sites. So we examined the writer and eraser because we are working on the upstream mechanism. So we excluded the eraser and focused on the writer and eraser only. And what we observed is intrinsic M6A site is significantly enriched with RBM15 and RBM15B, but the infrequent site doesn't. So the known RBM15 and RBM15B is one of the known accessory protein that helps to recruit methyl 14 and methyl 3 writer complex. So what we can see from here is RBM15 and RBM15B is likely recruit methyl 3 and methyl 14 complex to the intrinsic sites, but not the infrequent sites. So as maybe they can be methylated across all tissue types due to their help. So we actually examined the sequence level up to plus minus 150 base pair across all the intrinsic sites. And this one is...
Suming, I just want to clarify that. So when you identify RBM15 and 15B as the key writer proteins, are the supplement writer proteins to the methyl 3 and methyl 14. So this is what is the this bioinformatics approach that you took to
so
come up with this result.
So I retrieved the entire CLIFSEQ data from poster 3, which is the data set that the report data that showing the binding region of multiple RNA binding proteins, and we have multiple writer and eraser protein binding data in the poster 3, and then I examined the binding sensitivity in between 25 base pair on both, I believe. Not 25 base pair. I just examined everything and then compared and then analyzed the center of binding region from each site. And then the binding region was similar. It's all, pretty much like 12.5 base pair away from the intrinsic size or infrequent size, but their binding sensitivity was significantly higher near the intrinsic size only, but not the infrequent size.
So you examined a bunch of writers. How many writers that you examined here?
In the poster 3 DB writer, we have methyl 3, methyl 14, RBM15, 15B, and then Verma. And then I have one more, but I forgot the name, but pretty much 6 writers. And
okay, that cover probably most of the known writers for M6A, right?
But the issue is, I think we discussed this before, but methyl 3 and methyl 14 binding data is too small. It's not enough as RBM15.
Right. You already shown in the previous few slides that the RACH motif is significantly enriched. So, which is the binding motif of the methyl 3 and methyl 14 complex. So you can exclude methyl 3, methyl 14. So your goal here, so previously you find that methyl 3 and methyl 14 are the main complex, a writer complex, as in the case of all M6A methylation, right? And but the question is if the main methylation writers are the same as all M6A? And how do you explain that these so-called intrinsic sites are always being methylated across the tissue types? So their methylation mechanism should be unique. Right? So one of the hypotheses is that in addition to metal 3 metal 14 complex, there are unique complementary writers that are probably recruited by metal 3 metal 14 to function to methylate these tissue independent or intrinsic sites. So to kind of test this hypothesis, you investigate enrichment of bindings of bunch of writer complements or writer proteins, right? And namely these VERMA, RBM 15, 15-3, and so on and so forth. And out of all these writer proteins you investigate, out of these, I don't remember how many, 6 or 7 writer proteins that you investigated, which covers most of the known writer proteins for M6A, only RBM 15 and RBM 15B are enriched in the neighborhood of M60 sites. And this neighborhood actually is pretty tight, right? And specifically, you're showing this figure that the binding, the center of the binding is mostly enriched within 15 base pairs. Is that 15 base pair? 12 base pair. Yeah, 12 base pairs. It's suggesting indeed, they bind either right on top of M6A or in very close vicinity of the M6A enrichment, M6A methylation sites.
But yeah. So no other even erasers.
However, okay, no erasers. So no erasers are enriched. However, the binding of these writer erasers that you investigate are not enriched in any of the infrequent sites, right? That's which is shown on this panel on the right side, suggesting that RBM 15 or RBM 15B binding are unique to intrinsic sites, right? So that's kind of a support the hypothesis that you have. And also in the literature, it does show that RBM 15 and 15B are the supplemental writer proteins are recruited by metal 3 metal 14 complex, right? Or no, the other way around. RBM 15, RBM 15B binding recruits the metal 3 metal 14 complex, right? Yes. However, the context under which this recruitment happens has not been discussed. So here it seems to suggest that RBM 15, 15B, the binding RBM 15B is unique to intrinsic sites. That binding recruited the main methylation complex to methylate, to carry out this tissue independent or intrinsic methylation. Okay. Go ahead. Yeah.
So, so like Dr. Wang explained, all the literature is just showing very individual or independent experimental study that 15 and 15B is helped to recruit metal 3 and metal 14 complex to induce the methylation. So altogether, what we arguing is RBM 15 and RBM 15B is the key accessory protein that recruit metal transferase close to the intrinsic site so that they can methylate it context independently. And in our one of the feature of intrinsic sites was the sequence feature that neighboring sequence of intrinsic sites are different than infrequent or the background sequence. And if we examine the attribution score across the tissue, this is also analyzed by Ting He. And what we can see here is the positive attribution score, which means that these region of sequence are actually attributing to classifying these specific intrinsic sequence.
So means, sorry, I want to jump in just to, just to give a little bit of background why you're doing this, right? So, in figure F, you kind of show that RBM15 and 15Bs are the writer proteins that are unique to intrinsic sites. So they function to methylate M6A in this tissue-independent fashion, right? However, this doesn't explain why the methylation levels across different intrinsic sites are different.
Right? Oh, right.
So essentially,
so
if we have the same writer proteins shared by all intrinsic sites, right? And then this cannot be used to explain why the methylation levels are different. So the fact that the methylation levels are also different further suggests there are additional RE binding proteins, right? And they're probably working together with RBM15 and 15B to regulate the methylation levels.
Yes.
So the hypothesis here is that there are additional RE binding proteins, right? So to really look for these additional RE binding proteins. And
that might affect methylation levels.
Yeah. So you want to examine the neighboring nucleotides and to see whether there are unique motifs shared by different groups of these intrinsic sites. If there are, that probably suggests that a different group of intrinsic sites are bound by different RE binding proteins and therefore regulating different methylation sites. Right? So that's the underlying hypothesis, right?
That's correct. And
to do that, so you and Ting He used this classification, M6A classification model, right? And trained to classify M6 intrinsic sites from infrequent sites. Am I right? By using the neighboring, 100 or 120 base pairs up and down, upstream and downstream of M6A sites. Am I right? Trying to classify between intrinsic M6A versus infrequent M6A.
I believe this attribution score is intrinsic versus infrequent, not the background.
I know, that's what I was saying. Yeah, intrinsic versus infrequent M6A sites, right? Okay, so if the classification performance is high, that would suggest this nucleotide backgrounds between intrinsic sites and the infrequently methylated M6A sites are different. Right? So therefore, there are unique M6A motifs, nucleotide motifs in the neighborhood associated with intrinsic M6A sites, right? So I think here you need to show that performance, classification performance, just to support that indeed the nucleotide content up and downstream of M6A in intrinsic associated with intrinsic sites are different, significantly different.
Right? Yeah,
so the classification performance is what? Yeah. Okay. So you have like a 70 something, right? Okay. So suggesting there's a significant difference. Now, to pinpoint the regions where these nucleotide differences are between the two groups, between intrinsic and infrequent sites, you use the you use a deep learning interpretation method, specifically using what integral gradient to generate the so-called attribution scores for every individual nucleotide in this 125 nucleotide up and downstream of M6A sites. Right? So attribution scores indicate how relevant a nucleotide is in classifying these two groups of M6A sites. Right? The higher attribution score suggests the higher relevance of these nucleotides associated with one group or the other, right? So here you're showing so this plot G represent the attribution scores for every nucleotide that are important for intrinsic sites. Am I right? So, and you mainly observe two regions of interest, right? That spans, first is this green region, both locates upstream of M6. M6 is in the middle, right?
Yes.
That show higher attribution scores, right? And one is this blue region, the other one is the red region, right? Mm-hmm. And then you want to know whether there's unique motif or that associated with existing known RNA protein bindings, right? Mm-hmm. Or whether there are RNA protein binding sites that are also enriched in these two regions of interest. If there are, right, enriched meaning they're more enriched in associated with intrinsic sites versus the infrequently methylated sites, right? If there are, then it would suggest that these RNA binding proteins binds uniquely to this neighboring regions of intrinsic sites, right? And they are likely to be those RNA binding proteins that work together with RBM15 or 15B to co-regulate the methylation sites. Right. So, and then your figure H shows the enrichment of the binding sites of bunch of RNA binding proteins. Right. So you examine how many you examine, like 200 some RNA binding sites, binding sites of 200 some RNA bindings. So including a total of like 6 million binding sites or something like this, right? I think you need to speak, you need to tell people about these numbers just to show the amount of work and the breadth that you consider when it comes to the RNA binding proteins. And then specific, you identify what, now you can tell people, based on the results of age, what do you identify here?
So if I add a little bit more before we move on to the sequence, we kind of validated whether the methylation level is associated to RBM15 binding, but there was no association. That means intrinsic sites bound by RBM15 and 15B has higher methylation or lower methylation. It was random. So we kind of confirmed that this one is just helping for the recruiting the methyltransferase, but not affecting the methylation level. So we tried to figure out which one is helping to, or which one is affecting the methylation level. And then we wanted to see the other RBP bindings, like Dr. Ron explained. So we kind of grouped the intrinsic sites into the two. So lower methylation versus higher methylation cluster. And then between these two clusters, we examined this ROI1 first, because ROI2 is a 2 away from the M6A size. So it's a little bit difficult to say that how they likely affecting the methylation level. So we first examined the ROI1 first. And actually ROI2 has no significant differences when we analyze for the eraser part. But if we examine this ROI1 between this low methylation cluster and high methylation cluster, which is left side is high methylation cluster versus right side is the low methylation cluster.
So I mean, maybe you want to tell everybody why are you dividing the methylation sites in these two groups? What's your hypothesis?
Because we want to explain why they have different methylation level between the same intrinsic M6A sites. So our hypothesis is that there might be some other RBPs that affecting the methylation level between among the intrinsic sites. So we grouped into high methylation versus low methylation to expect to see the different binding between those high methylation, low methylation in these high attribution score region that is different. So this region is specifically different compared to the infrequent sites. And if this region or this sequence is associated to the methylation cluster, that means this specific sequence or motif feature compared to the infrequent site is affecting the methylation level among the intrinsic sites. But this RBM15 and 15B binding is around near this region that is also different than the intrinsic site, but it's not associated to the methylation level itself. So hypothesis was maybe there are the RBPs that more interacting with the eraser proteins, because we see that there are no difference between these blue dots are the erasers. So there are no different binding enrichment between the intrinsic versus the infrequent about the eraser, so that the nearby eraser binding is not affecting the methylation level or not affecting the binding between intrinsic or infrequent itself. So potentially this high attribution score or this unique sequence feature can be associated to the methylation level, especially in terms of binding with the eraser proteins. And indeed, we were able to identify the enriched RBPs that is specifically enriched in the low methylation cluster only, not the high methylation cluster. And they are known to interact with ALKBH5, which is eraser, or FTO, or both of them. So our conclusion is intrinsic sites have a binding enrichment in ROI1 is showing the low methylation among the intrinsic sites because of interaction between the erasers. So they are bind to this region and then recruit eraser protein near the intrinsic site, so as the methylation level is lower compared to other intrinsic sites.
Let me clarify this. I'm also a little bit confused here. Okay, I want to clarify just for my own sake as well. So here, okay, because we suspect that there will be unique RE binding proteins bind in this blue region. You call this region of interest 1, ROI1, right?
Mm-hmm.
And to regulate as a co-regulator of RE methylations. But more specifically, you would the hypothesis here is for M6A sites with different methylation levels, the RE binding proteins that bind in the neighborhood are probably going to be different. Right? So because of this, you group the M6A sites, it is intrinsic M6A sites, based on their methylation levels into two groups, namely the group with high methylation and the other with low methylation levels. And the hypothesis that the RE binding proteins in the low methylation enriched among the low methylation M6A sites might be different from the RNA binding proteins enriched in high methylation levels, right? So, or you aim to find these RNA binding proteins that are only enriched in one group of these intrinsic M6A sites versus the other group, right? So with this, you did the binding enrichment across this, 200 some RNA binding proteins, and what you find out, very interestingly and surprisingly, is that you only see a small list of RNA binding proteins highly enriched in the low methylation M6A subgroup, right, which is shown in figure H on the right side. Right, that's the group of M6A sites with low methylation, whereas the left panel of figure H is the enrichment of RNA binding proteins for the high methylation group, and where you can see there's nothing enriched, right? And a close examination of this list of enriched RNA binding proteins in the low methylation group identified the eraser protein, right?
No.
Which,
RBP interacting with eraser. They are not a known eraser.
Oh, okay. I see. Okay. So what you're showing here is out of this enriched among this for this group of enriched RNA binding proteins, enriched in the low methylation group,
are known to interact with eraser.
They are known to interact with erasers, and specifically. So the color of these RNA binding proteins show the specific erasers that are they interacted with, right? So okay, and then this result suggests that these low methylation group, these RNA binding proteins, they bind uniquely to the upstream of M6As with low methylation, right? And the reason why this group of M6A with low methylation is because they, all of them recruit erasers to reduce the methylation levels, right? Whereas for the high M6A group, we don't see any RNA binding protein enriched,
yes,
right? So they so suggesting their methylation levels are mainly regulated by RBM15 and 15B, and recruiting the main RNA methylation complex, methyl-3 and methyl-14. Right? So those low methylation M6A groups, their regulation of low methylation seems to be more complicated in this case, right?
So okay. In the very close vicinity, both erasers are not enriched near all the intrinsic sites, whether it's high or low methylation, but only these nearby regions. So probably this one is minus 20 to 30, 40. So it's gonna be here. So it's not that far away. Usually in between this region, this one is minus 50, so this peak is around minus 40, so upstream 40 base pair. So this region, the RBP enriched at this specific region, especially in the low methylation cluster, are the RBPs that are known to interact with erasers. That is, these are the data from the experiment, BioGrid experimental data, not even just the StringDB data. So this one is experimentally proved that they are known to interact with these erasers specifically. So what we can see is though the eraser binding are not enriched around the intrinsic sites, but the low methylation intrinsic sites have a neighboring sequence or neighboring binding RBPs that are known to recruit eraser protein close to the intrinsic site. So the likelihood of the demethylation process process among these low methylation cluster is higher than the this high methylation cluster. So as even though they are all methylated by the same methyltransferase complex helped by RBM15 and 15B, but this cluster is more likely to be demethylated due to this binding. That's the conclusion here. Any questions?
I mean, we have about 5 minutes left.
Yeah, so if not, let me
try see whether you can finish the next section in maybe 5-10 minutes, and then probably we can leave the rest of the sections for next time.
Okay, so let me try to finish the downstream in this minute. so far, what we observed is we identified intrinsic size, examined the interesting feature of those intrinsic size, and then we investigated why they are deposited across tissue independently, but also showing the various range of methylation level among the intrinsic sites themselves. And the next question is, what are the downstream mechanism so as why they are methylated and what happens after their methylation. So we are going to see the downstream result. So figure A is showing the methylation and the gene expression side by side. So what we can see here, the methylation and gene expression is significantly negative correlated, but also the gene expression is consistent across tissues, whether it could be high or it could be low. So the same analysis were performed for the gene expression so that the intrinsic size shows the significantly higher cosine similarity of gene expression across tissues compared to the background. Also show the significantly low standard deviation of gene expression across tissue types compared to the background. So what we can see here, there are tissue-independent gene expression and methylation, that are significantly consistent, but also significantly negatively correlated each other, suggesting that the key regulation function of intrinsic M6A size could be mostly the mRNA degradation. So what we observed, what we analyzed is, okay, then let's focus on the mRNA degradation function. And as I briefly introduced in the introduction or background part, DF1 to 3 are all known to work as reader protein to degrade mRNA. And also recently another paper is also showing, this one is 2023 paper, that is UPF1 also promotes rapid degradation of M6A containing mRNA. And I performed the same analysis using against the reader proteins and other RBPs, not the writers or eraser, what I did for the upstream. So this is downstream, so I focus on reader proteins only. And what we can see here, there are significant enrichment of intrinsic size compared to infrequent size, that of DF2, DF3, DF1, and UPF1 and PABPC4. So these three are the typically known are M6A reader protein that induce the M6 mRNA degradation. So intrinsic M6A size and their mRNA decay is possibly mediated by DF1 to 3 and the UPF1. And DF2 is the typical mRNA degradation protein, is the mostly significant enriched reader protein. And if we compare the combination of these DF1-3, DF2, and UPF1, that if we unify this binding of DF1 to 3 altogether, 82.5% of intrinsic sites are all covered by DF1 to 3. And if we intersect this DF2 and UPF1, then 56.9% were covered by this DF2 and both UPF1. So what we want to see is that whether these findings is actually significantly affecting the methylation and gene expression and their correlation. And we check the regression between the expression and methylation, comparing all the M6A sites versus sites bound by DF2, and also all the sites and bounded by UPF2 and DF2 and UPF1. And what we see here is bound by DF2 is has it's too small, but has a negative correlation between the gene expression and methylation, and also DF2 and DF1 binding also shows the more negative correlation than the average correlation. So overall, intrinsic M6A sites are bound by DF1 and UPF, YTH-DF1-3 and UPF1. And then they initiating the mRNA degradation at a downstream level. So time is almost up. So let me briefly focus on the enrichment result, or I was focusing
Do you want to maybe leave this for the next time?
Yeah, maybe I have a little bit more slides for the cancer report. So maybe I'll stop here. So I'll show you the last two parts in next time. Hopefully you guys have enjoyed the result. Or maybe if you have questions, let me know. I can answer it.
People question. Yeah, go ahead.
Yes, I do have a question. So one of the last things you showed was that the canonical, I might be using wrong words here, but like YTH-DF1 through 3 and then UPF1, that those motifs they're found to bind in the intrinsic, but not in the infrequent. What is how do you think, what's the interpretation of that?
So I haven't detailedly listing all the reader proteins, but there are many known reader proteins that has multiple different functions. Some is affecting splicing, some is affecting nuclear export, some is affecting the mRNA stabilization, and etc. And what we see from this binding significance between the intrinsic versus infrequent, and also you see this negative correlation between gene expression and methylation, what we conclude here is the main downstream function associated intrinsic sites is mainly the mRNA degradation compared to the infrequent site, because of these significant negative correlation, plus this significant binding sensitivity towards intrinsic site, especially DF1-3 and UPF1, which are all known to function to degrade the mRNA decay. And there are other enriched proteins are there, but if we compare to infrequent and gene expression data together, we are conclude that these could be the main downstream function associated to the intrinsic site.
I see. Okay, great. Thank you.
Yeah, I think what's really significant here is if you look at the enrichment ranking, figure E, YTH-DF2 ranked number 1, UPF1 ranked number 2, and YTH-DF3 and 1 are number 3 and 4. So Sumi has a really long list of the proteins. How many proteins do you have?
I examined 287 RBPs.
Okay, and it's really surprising and amazing that these reader proteins that are known to regulate, to mediate M6A, modular M6A mediated RNA degradation, all of them rank on the very top. So this is
It is
go ahead. Yeah.
Very validating for the understanding of the field, right? To say that you're kind of doing this in a very data-driven way, and you find exactly what these main readers are. So
Yeah. So essentially, the results suggest that these intrinsic side are not function to regulate, for example, translation efficiency, RA export, and so also splicing. And their main function is to regulate RNA degradation. So that's the message.
So
But Sumi, I have a quick question.
Mm.
So the YTHDF1, 2, and 3, they're known to be in cytoplasma, whereas UPF1 there is known to be inside the nucleus. So how do we potentially reconcile this?
So this recent paper is showing the UPF1's so UPF1 is in the nucleus, and their NMD RNA NMD is the separated ones. And this recent publication is showing that the UPF1 needs YTHDF2 initiated M6A mediated degradation. So I forgot the exact cellular location of UPF1, but they experimentally proved that the UPF1 require DF2 so that degrade the mRNA methylated transcript. So maybe UPF1 can function in other cellular location too. I can double check the paper.
Double check that paper and see how they address this issue, right? Okay.
But it's the figure is too small, but everything the center is DF2. So DF2 recruits something which recruit UPF1 that initiate degradation. And there are 3 different cofactors that are recruited by DF2 so that the recruit some other proteins to initiate the degradation. So I think this one is pretty recent paper, so maybe there could be some follow-up publication afterwards. So I'll double check on this. I guess there are no more questions. Okay.
Do we have more questions? No. Okay.
I don't think so. Do we have? No? Okay.
Okay, Sumi, so we'll give you some time next week at the beginning of the lab meeting to have you finish the rest of 2 sections.
Oh, next week?
Okay. Yeah. Right? Okay.
Okay.
So Hugh is also scheduled to talk next week.
Okay. And then Hugh can follow up on his presentation. Yeah. Okay. All right. Thank you, everybody. Thanks, Sumi. Yeah.
Thank you, Sumi.
Good
Thank you, Sumi.
presentation. Yeah. Bye.